Deu, ok.
It's our pleasure to have Ismael Galve Roperth talking to us today. And he's already a friend from Spain that has been in Brazil more than once now, right? Much more than once. And he's an associate professor at Complutense University in Madrid, Spain, and he leads a research group in cannabinoids and neurogenesis, I would say in development of the CNS. And he has been investigating or studying the role of cannabinoid signaling in brain development and Huntington's disease associated neurodegeneration. And I think it's interesting and of note that he has been a pioneer in demonstrating the anti-tumoral action of cannabinoid administration on glioma brain tumors. And I think that also he works in the endocannabinoid system and stem cell-derived neurogenesis and the impact of prenatal THC exposure and brain function. But Ismael has also been a founder of the Ibero-American Cannabinoid Research Network, CANALATAM, CITED, which he coordinates, so he probably will say something about this later on. Thank you so much, Ismael, for your acceptance of talking to us.
Thank you, Cecilia. So first, thanks, Cecilia, for contacting me and inviting me to this presentation. I will go ahead in English with the presentation, but in any case, I understand Portuguese, so please do not hesitate to interrupt me if I'm going too fast. The online thing is kind of difficult sometimes to get the feedback from the audience, so please do not hesitate to raise your hands through the chat if I haven't seen it, or just say something on the microphone, connect your microphone, and you can interrupt me. There is no problem with that for me. So my presentation today is about the role of cannabinoid signaling in neurodevelopment. I'm going to be discussing different things related to what's the role of endocannabinoid signaling, the endogenous system, which is going to be crucial in the development and the maturation of the nervous system, either during embryonic development and later in the adolescence. But also, this field has important implications in terms of health and disease. So we will see how cannabinoids are proposed to exert some anticonvulsant actions in, for instance, refractory epilepsy. We will also address briefly the implications of prenatal cannabinoid exposure in the behavior of the progeny, and I will go through some of the experimental data that we have generated in the lab. So I believe most of the people in the audience will already know that cannabinoid signaling is mediated by membrane-located receptors. Those receptors are called CB1 receptors, and these receptors are modulated by either exogenous exposure of plant-derived cannabinoid, namely THC. So THC is going to be the main active compound of marijuana, acting through CB1 receptors, which we don't synthesize them in our organism to be able to respond to THC. The reasons why we have these CB1 receptors is because they are targeted by endogenous molecules. Cannabinoid signaling is mediated by...
Espera aqui. Loud. Só um pouquinho. Só um pouquinho.
Can I go through?
Yeah.
Okay. Yeah. So we do not express CB1 receptors for the THC, but we do it because we have endogenous ligands, the so-called endocannabinoids. The most important are anandamide and 2-arachidonic glycerol. And as you can see them, they are lipid-derived structures, so they are ethyl ethanolamides derived from arachidonic acid. What's the main function of the endogenous system, of the endogenous cannabinoid system? It's neuromodulation. endocannabinoids are released in synaptic clefts after neurotransmitter release. So in many kind of synapses, glutamatergic synapses, GABAergic, many kind of them, neurotransmitter release engage postsynaptic receptors that are going to be able to prompt endocannabinoid release. So endocannabinoids are released at the postsynaptic level in an activity-dependent manner, and this is going to release the endocannabinoids to the synaptic cleft. They will go backwards, and they will travel backwards to in a retrograde manner to the presynapses to target those CB1 cannabinoid receptors. And it's not a trivial thing that the CB1 receptor is one of the most abundant G protein coupled receptors in the brain. So it's really very highly expressed in the nervous system. It's present in many synapses. And then either endocannabinoid signaling influences many functions of the brain. This neuromodulatory role of the endocannabinoid signaling system explains the contribution of this system to cognition, to learning and memory, for instance, to the regulation of behavior and emotions, such as anxiety or depression, the regulation of excitotoxicity and epilepsy, for instance. The diversity of endocannabinoids is pretty big. We have many candidate molecules that are proposed to target cannabinoid receptors CB1, although the most important are going to be 2-AG and anandamide. And this is the distribution of the cannabinoid receptor, the CB1 receptor, which is a G protein coupled receptor. As you can see here, this is an in situ hybridization or a binding, I don't remember well, about showing in coronal sections the expression of CB1 receptors in wild-type mice. You can see there is a very high expression in the striatum and the globus pallidus, in the hippocampus or in the cerebellum. And this explains that the CB1 receptor is going to be expressed both in glutamatergic projection neurons, but also in interneurons. And actually, even if the CB1 receptor is expressed at much higher levels in interneurons, the low levels of CB1 receptors in glutamatergic neurons have also very relevant functions in the regulation of physiology of the nervous system. But all this functional role of the endocannabinoid system relates to the adult brain. There was a lot of interest in knowing and understanding what was the mechanism of action of THC, of this plant-derived cannabinoid, and they discovered, and the search for the molecular targets of THC resulted in the discovery of the endocannabinoid system. So when scientists were looking for the mechanism of action of THC, at the beginning it was thought it was through membrane distortion of membrane fluidity. Later on, the receptor CB1 and CB2 were cloned, and then also the endocannabinoids were discovered. And in this field, there was a lot of interest to investigate what was the consequences of THC exposure during the adolescence. So there have been many studies using pharmacological manipulation in embryonic development or in postnatal development. And researchers were looking on the progeny later on, and they could find how THC exposure was inducing psychiatric traits, influencing anxiety, for instance, social interaction, also motor behaviors. The endocannabinoid system was, manipulation of the endocannabinoid system through cannabinoid exposure was affecting also excitation and inhibition balance. So yielding to a circuitry which is more prone to convulsions and seizures, or also THC exposure was resulting in altered addiction susceptibility. So it becomes pretty clear that during embryonic development and adolescence, there is many active things happening at the cellular level, which are much more active than in the adult brain. So during embryonic development and later on, we will have very active processes of cell proliferation and generation of neurons. We will have the neuronal differentiation and migration to the cells that they need to reach their final place. And then we will have the final network establishment and synaptogenesis to develop functional circuits. Hence, during these sensitive periods, either malfunctioning of the endocannabinoid system due to genetic polymorphisms or environmental exposure, for instance, due to prenatal THC exposure, is going to be able to modify many of these processes, resulting in altered wiring of the brain, which make it more susceptible for developing neurological disorders later on. So are cannabinoids a bad thing or a good thing? And here the field has been moving, and we will find many controversial opinions. We have moving the pendulum from a perspective where cannabinoids and marijuana was like the assassin of youth, and it was an evil molecule or an evil drug. And from here, we have maybe moved too fast also to think that cannabinoids have a broad therapeutic potential that they can cure almost any kind of diseases. And of course, this is not going to be true. There will be some applications and many others that will fail and there will be able to be demonstrated. For instance, we know due to this neuromodulatory role that CB1 receptors are important controlling convulsions and epilepsy. We know, for instance, that CB1 receptor blockade increases the severity of convulsions. As you can see here, this is a EEG recording of an epileptic rat. And when we block cannabinoid receptors, the severity of the convulsion of the seizures is much higher. We also know that genetic ablation of CB1 receptors in preclinical models give rise to a more sensitive neuronal circuitry to convulsions and seizures. And this is due to the expression of CB1 receptors both in pyramidal neurons, shown here in excitatory projection neurons, but also in inhibitory CCK basket cells. So indeed, cannabinoid receptors are very well located to be acting as a master regulators of the homeostasis and the excitation and inhibitory balance. Actually, if we analyze post-mortem samples from patients from temporal lobe epilepsy, there is a reduction on CB1 receptor levels, but also on endocannabinoid levels. So there is a current hypothesis that downregulation or malfunctioning of the endocannabinoid systems may underlie some kinds of epilepsy and convulsions. And actually, the administration of different plant-derived cannabinoids exert, as shown here in green, anticonvulsant action. So mainly the activation or the regulation of the endocannabinoid system, but also using CB1 receptor agonists results in anticonvulsant actions. As you can see here, THC, delta-9 THC, but also CBD, which is another cannabinoid, and is not acting actually for CB1 receptor, exerts anticonvulsant actions. So we have a very complex pathway. We have THC-mediated actions, which are dependent on CB1 receptors. We know that activation of CB1 receptors exerts an anticonvulsant action, most of the cases. It may exert proconvulsant action when cannabinoid THC is inducing the downregulation of the receptor. But also, THC exerts psychotomimetic action, so it's a propsychotic compound, and is also an important modulator of emotions. So this makes that the use of CB1 modulation as an anticonvulsant action has been very difficult to be demonstrated, and the efficiency of these strategies under question. It's not yet achieved. However, as I mentioned, CBD exerts actions independently of CB1, and they are anticonvulsants, and even more, CBD is a molecule which exerts antipsychotic actions and also anxiolytic actions. So actually, THC and CBD act in opposite manners and they counterbalance each other. Moreover, although the field is not yet there, in addition to use natural compounds derived from the plant, of course, the pharmacology of the system has a lot of possibilities. There's a lot of synthetic cannabinoid ligands either targeting CB1 receptors or CB2 receptors that are under development. The use also of metabolism inhibitors, and this is going to be important. We have already second and third generation of inhibitors of the degradation of anandamide or 2-AG. For instance, those compounds shown here are inhibitors of the degradation of 2-AG. The use of these compounds allows to target CB1 receptors exclusively on the synapses that are active because the endocannabinoid tone is actively engaged. So the use of these metabolism inhibitors has a very interesting profile because it allows to modulate the cannabinoid receptors in an active dependent manner and not in a broad action along the whole CNS. And so this is the efficiency of CBD as an anti-epileptic. EPILEPSY is the approved pharmaceutical presentation for this. This is developed by Galve. And as you can see here, CBD has been shown in double-blind clinical trials, so in standardized clinical trials. This is actually not the rule in the cannabinoid field. There are many anecdotal evidences of cannabinoid actions in therapeutic aspects, but in most of the cases, clinical trials are very scarce in number of patients. They are non-controlled or they don't have placebo controls. So CBD has been one of the exceptions for that. There's been very good clinical trials developed. And as you can see here, for instance, CBD is able to reduce by more than 40% the convulsions in patients with Dravet syndrome, the Dravet syndrome, as well as other refractory epilepsies like Lennox-Gastaut, tuberous sclerosis, or febrile infection-related epilepsy. So actually, CBD has exploded as a new tool as an anticonvulsant action, even though we really do not know yet which is its mechanism of action. Actually, even if CBD is a very interesting molecule in terms of neurological disorders, it's also proposed that it may act as a molecule acting on neuropsychiatric disorders such as autism and Down syndrome. So a mixture of high CBD extracts and low THC containing extracts, so compound extracts that have a lot of CBD preventing the psychotic actions, and some THC agonists have been tried to treat patients with in children with autism. And actually, in clinical trials, some people is claiming, according to the caregivers and the perception of the caregivers, that there is some therapeutic options for cannabinoid management of autism. Of course, there is a strong concern about what are the deleterious consequences of THC exposure during the embryonic development. It is pretty well known that embryonic THC may exert a second hit stressor and a stimulator for the development of psychosis and schizophrenia. And in this GWAS analysis, this was the biggest GWAS analysis performed so far in order to analyze the influence of cannabinoid exposure during development and the development of schizophrenia. And in this analysis, which involved 200,000 persons, it was shown that actually there was a parallelism between consumption of cannabis and genetic traits that prone to schizophrenia, bipolar disorder, or autism. So actually, in this report, rather than stating that THC exposure is a causal stressor leading to schizophrenia, actually it may be the opposite. It may be that patients are self-medicating because they already have a circuit which is making them more prone to these neuropsychiatric disorders. So actually scientific information in this field has to be taken in very, taking a lot of attention to how science is described and how the interpretations are provided. So I would like to maybe do a small pause. I don't know if someone wants to comment so far about the talk, or if I should follow up. If I'm going too fast, everything is feeling okay. Everybody's feeling fine.
I think it's fine. It's going
Okay.
good.
Not too fast,
No, it's fine.
not too dense for the audience. Okay, so we will move. I'm gonna move now to some research that we have done regarding what's the role of the endogenous cannabinoid system in prenatal development of the nervous system. As you can see here, we are going to have CB1 receptors that are going to be present during embryonic development. There are also active production of endocannabinoids during embryonic development. And from the findings from our research and other labs, we know that cannabinoids are able to regulate progenitor cell proliferation and identity, neuronal differentiation, and also neuronal migration. So cannabinoids, the endogenous cannabinoid system is present during embryonic development, and this is important in order to understand the consequences of exposure to cannabinoid during development. So one of the stories that I'm going to let you know today is some experiment that we were doing manipulating cannabinoid signaling during embryonic development. So the idea here was to downregulate cannabinoid receptors. So we use an approach called in utero electroporation, in which we can manipulate injecting a small interfering RNA for the receptor at embryonic development in the pubs of a pregnant mice. CB1 receptor is going to be downregulated during embryonic development, but later on, when adolescence and neuronal activity has taken place, CB1 receptors will be present back to the circuitry. And then we are analyzing the consequences of the neurological function and behavioral function of these mice. The idea, as I was trying to point it out, is that we are going to be investigating the consequences of cannabinoid exposure in the adult, and the changes that we are observing here are going to be due only to the absence of CB1 receptor during an embryonic development. So during a moment when there is no neuronal activity or no synapse-driven neuronal activity. And this is important because otherwise we will be looking to many changes here due to the modulation and adaptation of neuronal activity. So when we were doing these experiments, we inject CB1 receptor siRNA, so we knock down the receptor, and we label the cells with a green fluorescent protein, as shown here. And as you can see here, you can see on the lower part of the slide much better. CB1 receptor was downregulated at embryonic day 14. Neural progenitors are proliferating in the ventricular and subventricular zone. They will exit cell cycle, and they will differentiate along the cortical plate. So in the absence of CB1 receptors, we found there were ectopic neurons that did not reach the final location. We found neurons that remain located in lower areas of the cortex instead of being in the final page where they should be. And even more, we found cells stuck in the subcortical white matter. So there were this was like a migration phenotype, migration arrest phenotype induced by the absence of CB1 receptors. Indeed, if we look later on and we go up to 60, 90 days, so these are adult mice, again, those adult mice that were downregulated the receptor solely and only during embryonic development have neurons, ectopic neurons in the subcortical white matter, and these neurons apparently did not have changes in the differentiation. We haven't investigated too much about that, but the thing is this phenotype looks like pretty much to an epileptic syndrome called subcortical band heterotopia. So this raises the question of whether there will be endogenous mutations of polymorphisms of the endogenous cannabinoid system that may relate or may be the cause of some pediatric or malformations of cortical development. So if we now don't regulate the receptor and there is a blockade of neuronal migration, is there any role for the endogenous ligands acting as chemoattractant signals? To investigate so, what we did is we exposed developing neurons. So we exposed the cortical explant from embryonic mice. to a source of endogenous cannabinoids. So we transfected cells with a plasmid that allows these cells, these COS7 cells, to express either anandamide or 2-AG. And then we will analyze how the neurons come out from the explant and migrate towards the source of the signal. And when we did those experiments, as shown here, we are quantifying the neurons that migrate towards the proximal side from the source, and the distal side, the opposite side. So as you can see here, when the grain cells, the COS cells, are expressing 2-AG, they're releasing high amounts of 2-AG, neurons migrate towards the source. Same case occurs with anandamide, so cells expressing anandamide attract the neurons towards the source. And these actions are CB1 dependent because we can block the migration if we block the receptor pharmacology with a CB1 antagonist. So the CB1 antagonist blocks neuronal migration induced by endocannabinoids. And actually, during development, if we delete or if we abolish the endocannabinoid gradient, we can overexpress the enzyme that is responsible of the synthesis of 2-AG. So we are overexpressing the enzyme of 2-AG here. We are increasing the levels of 2-AG, and now there is no the gradient anymore towards the upper side. And then this makes cells again to remain blocked in the subventricular and ventricular zone. So is there any correlation between these neuronal migration deficits and cortical excitability and convulsions? To investigate so, we approached to a mechanism by which we were able to reverse the cellular consequences of CB1 downregulation. So based on biochemical findings of how CB1 receptors were acting, we knew that CB1 receptors were acting through the small G protein rho A. And as you can see here, we can restore the neuronal migration phenotype, or the ectopic neurons, due to the ablation of CB1 receptors by knocking down the rho A protein. So we ablate the receptor, neurons migrate, neuron migration is blocked, then they remain stopped, and we can revert these phenotypes by manipulating another signaling system. What happened in terms of convulsion on these mice? Knocking down the receptor solely during embryonic development, so exclusively during embryonic development, results in mice that are more prone to convulsions. These mice have more sensitivity to seizures induced by a proconvulsant drug. And if we revert the neuronal migration phenotype induced by CB1 receptor downregulation, we also revert the proconvulsive phenotype. So actually, this is showing that there is a cause relation between CB1 receptor downregulation during embryonic development and the development of seizures. One of the major conceptual implications of these findings, it means that cannabinoid signaling, endogenous cannabinoid signals, are important in the regulation of neural cell fate during development, and those actions are in addition to the neuromodulatory role of cannabinoid signaling. So cannabinoid signaling, of course, most of its action in the adult are going to be due to neuromodulation, but during development, they are important modulators of cell fate, differentiation, and or migration. For instance, we know that when we're knocking these receptors during embryonic development, there are changes in the maturation of neuronal maturation. So neurons that are developing during embryonic development and that we knock down the receptor, they have changes in the acquisition of the neuronal excitability properties, both active and passive properties. And so one of the questions we were wondering is whether there's any implications for cases of human epilepsies. So cases in refractory epilepsy where mutations are associated to neuronal phenotypes that are related to this, to similar things that we have found in preclinical models. So, for instance, there are a whole panel of diseases called enteropathies, in which there are mutations of the PI3 kinase, AKT, or the mTORC1 complex, and some of them include focal cortical dysplasia, hemimegalencephalia, or tuberous sclerosis complex. So we analyzed what was the expression of the endocannabinoid system in some of the diseases, because we also know that CB1 receptors are present during human cortical development, and this was a previous report showing that in focal cortical dysplasia there is a high expression level of CB1 receptors. So we move forward and we analyzed a cohort of 20 focal cortical dysplasia patients together with 10 controls. We were analyzing samples that are coming from the children that have this focal cortical dysplasia, and they are subject to surgery to remove the source of this intractable epilepsy. We did so, and we analyzed mRNA levels by qPCR. We also analyzed gene polymorphisms for different elements of the endocannabinoid system. And what we found that there were 3 polymorphisms of the diethylglycerol lipase enzyme. So patients with focal cortical dysplasia showed different polymorphisms for the enzyme responsible of the synthesis of 2-AG. And actually, we observed in the patients that there were higher levels of mRNA for CB1 and lower levels of the enzyme responsible of the synthesis of 2-AG. So this suggests that somehow alteration of endocannabinoid signaling can have a role on the development of this kind of pediatric epilepsy. And of course, one of the, let me see the time, yeah. Of course, one of the important implications also of investigating the role the endocannabinoid system during development is to understand what are the consequences of pregnant mother exposure to THC. So, of course, this is an oversimplification. This is only an animal model. And I always say when I'm mentioning these studies that we should take care, we should be very careful with the interpretation of the data. We cannot really mimic exposure or modeling developmental THC exposure. One of the main objections I will propose to my experience is we are injecting a single molecule. We are only injecting THC, which is not going to be the case of human exposure. So human exposure will be cannabinoid extract containing not only THC but other molecules, mainly CBD. So probably the effects are going to be different. However, let's investigate what's the consequences of THC exposure during embryonic development. So in order to do so, we used pregnant female, which were CB1 knockout mice. So these pregnant female were crossed with a CB1 heterozygous male. So the progeny is going to be 50% plus minus and 50% minus minus. We are going to have a litter mates here that are going to be analyzed on the progeny, on the adulthood. They are treated with THC during 5 days on an embryonic dose of 3 milligrams per kilo, which is not a very big amount of THC. It's kind of like the dose, an average dose equivalent to a joint. And the THC should be able to cross the blood-brain barrier from the mom and reach the pups. So we are having a control within the experiment because we can analyze the consequences of THC in the progeny of this pregnant female that has no CB1 receptors, so THC should be inactive on those descendants, and we also will have the other half of the litter mates will have a receptor copy, and they will be sensitive to THC. And we analyzed it after these 5 days of embryonic exposure, the consequences in terms of behavior, excitability, and neuronal alterations. What we found in this experience was the following. Those are adult mice exposed to a proconvulsant drug used as a readout of sensitive to convulsions and seizures. And as you can see here, only the absence of CB1 receptor, which is the blue column, makes these mice more susceptible to convulsions. And this is always been attributed to the fact that these mice have lost one of the main neuromodulatory systems, which is presynaptic CB1 receptors. But in addition, heterozygous mice exposed to THC solely during embryonic development, only 5 days, they haven't seen THC later on for more than 2 months, these mice still have more convulsions to see, more sensitivity to convulsions. And again, or not again, and as a control, CB1 knockout mice, they are not sensitive to THC. So actually, THC during embryonic development via CB1 receptors is doing some plasticity events that result in a more sensitive circuitry. And actually, this is due to downregulation of the receptor. So transient downregulation of the receptor during embryonic development, so the same phenotype than the knockout mice, give rise to this pro-epileptic phenotype. Even more, we found that exposure to THC resulted in alterations of 2 main populations of the nervous system, both projection neurons and interneurons. As shown here, mice exposed to THC during embryonic development had lower number of neurons called deep layer neurons. So the neurons that are located in deep layers of the cortex, and they are used to control corticospinal function. So these neurons are projecting from the cortex to the spinal cord, and they are going to be important in some motor behaviors which relate to skilled motor function. In order to elucidate in an unequivocal manner what was the role of CB1 receptors, we use transgenic knockout mice in which we have a genetic background which is a stop CB1 background. So these mice do not express the receptor and they are as knockout mice. What we can do is, through the use of the Cre recombinase technology, we can re-express the receptor CB1 by deleting the stop signal, which is located in front of it, before it, in glutamatergic neurons, in interneurons, or in the whole organism. So this is a expression rescue strategy. And if we do so, and here is the proof of evidence that indeed we are able to rescue expression, wild-type mice express the receptor, stop mice, they behave like knockout mice, and full CB1 rescue or glutamatergic neurons allow the expression, the re-expression of CB1 in glutamatergic neurons, or in all neurons, or in interneurons. Okay, so what happens when we do so? When we do so, we can observe here that the expression of CB1 receptors in either pyramidal neurons or re-expression in interneurons rescue the sensitivity to seizures. Hence, both CB1 receptors present in interneurons and pyramidal neurons are required or contribute to the pro-epileptic phenotype. What happens when we treat with THC? THC is not able to induce more seizures in mice that do not have CB1 receptors, null mice. However, THC is able to increase THC sensitivity to seizures in any of the rescue mice. So actually, we can demonstrate that CB1 receptor present during development in either pyramidal neurons or interneurons are mediating THC actions. One important thing that we found in our project was that when we were analyzing this epileptic phenotype, we found that we were mixing the mice according, without taking account of the sex. And this was giving us a lot of problems to find statistical significance. And we found that there was a divergent comportment of the mice. And actually, as you can observe here, most of the mice that have developed sensitivity to seizures are the male progeny, but not the female. So actually, the male progeny of the pregnant mother is actually the cohort that is more affected by THC exposure. So this is giving us some information that there are sex-dependent differences between the consequences of THC exposure during embryonic development. I was mentioning that THC not only modifies pyramidal neurons, but also interneurons, and actually interneurons, these are hippocampal CCK basket cells, measured in both males and females, and as you can observe here, the phenotype is selective for the male progeny. So THC exposure during prenatal development induces an interneuronopathy in the hippocampus and in the cortex that may be responsible of the epileptic phenotype. In addition, the consequences of THC exposure expand also to cognitive function, and prenatal THC exposure interferes with spatial memory, as shown here by an analysis called novel object location, but it doesn't interfere with general novel object recognition function. So prenatal THC alters some motor functions by interfering with corticospinal motor neurons and interferes with cognitive function and the excitatory inhibitory balance by altering interneuron development and specifically CCK basket cells. And finally, one of the things we wanted to analyze in further detail was the influence of THC in the whole synchrony of the circuit. So what's the consequence of prenatal THC exposure on the oscillations and the synchrony of the circuit? And we did so by using multi-electrode recordings, intra-hippocampal multi-electrode recordings with in vivo mice running on a wheel. And as you can see here, the male progeny exposed to THC had lower theta frequency. However, the gamma frequency was not altered in the progeny of the male. So we observed theta changes in the male progeny, and also we found changes in high-frequency oscillations. These high-frequency oscillations, or also called sharp wave ripples, are very important markers of the coordination of the circuit. And as you can see here, in the male progeny, there was an increase in the lower frequency of these sharp wave ripples. So there was an increased number of these smaller sharp wave ripples in THC-treated mice. So in general, we can say that manipulation of cannabinoid signaling has the potential to modulate, either improving or worsening different neurodevelopmental disorder symptoms. There is an important need of clinical trials and a better understanding of the mechanism of action of cannabinoid signaling during development. We cannot say because they are different, phytocannabinoids are different. THC is a complete different molecule than CBD, and they don't act by the same mechanism, and they have different consequences. It may be think that in the future, in addition to the use of phytocannabinoids, which are plant-derived, we can move forward to the use of synthetic cannabinoids. They still have a long road ahead, but this is probably the future of the field. And about our contribution to the field and our contribution to the knowledge of cannabinoid actions, we would like to propose that cannabinoids are important in neural development, not only modulating neural activity, but also modulating neural cell fate, so modulating the number of neurons that are generated or how these cells are differentiated and establish connectivity. And my last conclusion will be, we need rigorous science to analyze the development of cannabinoid-based medicines and taking a safe risk assessment of its recreational use. And I would like to thank also, of course, the people of the lab that has done the experiments that I have shown here. Many people and very hard workers and clever guys have been investigating these things. Some of the people are here, current members of the lab and also previous members of the lab. And of course, I would like to thank our collaborators in different labs worldwide, Germany, Madrid, and Spain, also in Brazil, in Sao Paulo. We have strong collaboration, and of course the funders that provide us the money and the funding to allow to do our experiments. And I would like to thank you for your attention. I'll be happy to hear your questions.
Okay, thank you, Ismael. So can you tell us a little bit of how Canalata is going to function? What are the aims for the network? And
okay,
how
maybe you can go first before the people forget about what I mentioned.
Okay.
I don't know. Maybe I don't know what if there is questions. If there is no question, I can.
There are questions. There are questions.
Okay, so let's maybe if you agree, we can do first. We can do that first, and then okay. How are we gonna concern?
Thank you. I'll ask Rafael. Rafael is already put 2 questions here. Do you want to speak, Rafael? You want me to?
I can't speak myself. Can you hear me?
Yes, thank you.
Oh, okay. So I had actually 2 questions. One refers to the CB1 knockout animals. Can you discriminate effects directly on the neuronal synapses or indirectly via different glial cell types? Or is there any data that that can give a hint on specific effects of each one of these CB1 expressing cells in the effects that you have in your? I assume that you are knocking down the receptors on all cells in these animals.
Right. So this is actually a very good question. So. Actually, as you mentioned, the complete CB1 knockout mice is a full CB1 knockout mice, and of course, those results are very broad in terms of the meaning, right? We know there's a lot of compensation on those mice, and so the phenotype that we are observing is a very indirect phenotype at the end of all these circuits. We've been using next CB1 deficient mice, for instance, and interneuron deficient mice. And we know that those mice have changes in neuronal development when CB1 is only ablated in projection neurons or interneurons. Regarding other cells, CB1 is present during astroglial differentiation and probably also during myelination of oligodendrocytes. However, we haven't been investigating the selective non-cell autonomous component of this action. So most of the things we've been investigating is cell autonomous action on neuronal differentiation or development. But that's a very good question, indeed.
Okay, thanks. I have one more. Can I go on, Cecilia?
Yes, you can.
Okay. Is there any evidence that CB receptors interact with TRP-type channels?
So you mean heteromers between TRPV receptors and metabotropic receptors?
Yes.
There may be, and we can probably find in the literature some examples of that. I like more the idea of the CB1 homodimers or heterodimers with CB1 and CB2 receptors, but I do not discard that those heteromers with ionotropic receptors, they may exist. But the thing is, it's very as you're very aware, membrane, receptor oligomerization is a very complex field yet, and it still has a long way to go. The proximity ligand assays is complex to be done, and the resolution is not very well. It's not very solid. So, but yeah, that's plausible.
Okay, thank you again. It was a wonderful talk.
Thanks for your questions, Rafael.
Lucia, do you want to make your question, or you want me to? Are you there? Lucia put a question of whether the administration of THC is regulating other cannabinoid receptors like CB2 and GPR55 or TRPV1.
So whether THC does so? If it does regulate those other receptors? Probably it does. Probably it does. We've been focusing only on CB1. To say the truth, yeah. As I was telling to Rafael, probably neuromodulatory actions on CB2 receptors are also very important on the consequences, mainly probably on adolescents. So cannabinoid exposure during adolescence is going to be interfering not only with the refinement of the circuitry, but also on myelination through the oligodendrocyte side and also on the inflammatory status, for sure.
Right. And she also asks whether you were able to measure the endocannabinoid levels of anandamide and 2-HG.
In development?
Yes.
No, we have done it in vitro. So we've been able to demonstrate in vitro, for instance, that neural progenitors produce in a cell-autonomous manner endocannabinoids. And actually, I can give you a hot new. This new paper from our lab just published in Development showing CB1 receptor regulation of neuronal differentiation in a cell-autonomous manner. So this is just came out in Development this week.
Oh, wow.
But we haven't investigated the role of neuroinflammation influencing neuronal differentiation.
I think she was asking whether the levels of the endocannabinoids change with the THC administration. But.
Okay. So that we haven't done. The thing is, it's not easy to measure endocannabinoids in such a small brains. So.
Oh yeah, that's right. Okay, Marilia, go on with your question.
Thank you, Ismael. Excellent talk. I have two questions, but one first, and then there's another one waiting on the line. I was wondering if you looked at the hypothesis that that GABA goes from excitatory to inhibitory during development, right. And some people think that in epilepsy GABA just remains a little excitatory, that's even after birth and everything. So I was wondering if you looked at how GABA is behaving in terms of excitation.
We haven't done it, but there's another group. There's another group in Marseille, in France. Olivier Manzoni is the director, the PI, and they have shown indeed that these changes in. Then they claim that this change. There's a delay on the switch of the NKCC1 and KCC2 channels for the GABA, and this will keep GABA excitatory for more time.
But this is induced by THC, no?
Yeah, this is by THC. And this is one of the proposals. So one of the things here is like, we can approach to the questions on experiments injecting THC, but I also believe that we need to know a lot looking solely on the endogenous system. So for me, any manipulation with THC is fine, but we need then later on to move to the biology and understand what's the precise role of CB1 in different neuronal populations. But yes, there is many indications for that.
Anyway, you're using pure THC.
Very good question so far.
You're using pure THC, right?
We are using pure THC, 99%, yeah.
Okay, so now there's Agustin has a question. Are you there, Agustin? Well, okay, so you can...
Oi, boa tarde. Boa tarde, Ismael. Não, minha pergunta é diretamente sobre o papel da exposição do sistema canabinoide na embriogênese tardia e pós-natal na diferenciação daquelas progenitoras neurais na zona subventricular. Se vocês têm observado, além da diferenciação, migração neural, alguma coisa de oligodendrogênese, gênesis ou astrogênese. Obrigado.
Então, no desenvolvimento pós-natal, tem estudos que demonstram que canabinoides eles modulam a mielinização, não é, dos oligodendrocitos. Nós vimos com outro grupo também o papel dos receptores CB2 nos progenitores da SBZ, da zona subventricular. Então, os neurônios da zona subventricular, eles vão no bulbo olfatório normalmente, mas em situação de dano, de neurodegeneração, eles podem ir no córtex, quando tem isquemia, ou no estriado, se tem enfermidades degenerativas, não é? Então, a gente demonstrou que tem um papel do receptor CB2 na migração das neurônios da SBZ à corteza no modelo de stroke.
Ah, legal.
Thank you. Tem Newton, você está aí?
Yes, I can ask if you like.
Okay.
Oh.
Go ahead.
Ismael, thank you very much for the wonderful talk. I'm very curious about your ideas on the basis of the gender effects in everything that you measured there. Do you have any neurochemical hypothesis for what's happening there that makes females different from males?
Mm-hmm. I wish I had. No, we haven't found any real substrate for this difference. I mean, it's pretty well known that the developing brain has gender or sex differences between males and females. So my feeling is that it's something related to the hormone status, for instance, the pregnenolone or levels of other steroids that may be influencing receptor downregulation and functionality. But we haven't been able to find any difference. We've been addressing if there were differences between CB1 downregulation in the males and the females. We didn't find any difference. We try to measure endocannabinoid levels, and we couldn't determine differences because the levels were very low. So it's not an easy task, but I'm sure it's the hormonal. I'm pretty sure there must be some changes in the hormonal niche that they are making the difference.
Okay. I was wondering whether, since you showed that there is a interneuropathy specific, maybe it's just the neurosteroid levels that would build on top of the difference in interneuron numbers.
So what was again your proposal?
Well, maybe it is the endogenous neurosteroid levels, the hormone-based neurosteroids, but then it's because of the difference in sensitivity, perhaps due to the difference in interneuron numbers, that would make the gender difference on top of that.
But then the question is why we don't have interneurons changes solely in the males. We still have, so we still have to find. Right. Yeah, but I get it.
It's circular. It's circular.
Yeah, it's a circular question.
Okay. Thank you. Thank you, Vena.
Thank you. Claudia, do you have any questions in YouTube?
No question. No question.
I have one more.
Oh, Maria, go ahead.
It's a quick one. I was also wondering if you try to reverse the effect of THC in down regulating CB1 by using CBD to restore the levels of CB1. Do you think it would work?
Probably, I believe it will work. So I believe that we believe that the consequences of THC is more because it's, even if we don't use a big dose, it induces somehow the desensitization of the receptor during some moments which is required. My gut feeling is that CBD is going to attenuate those actions. The thing is, for me, it's like those are very, let's say, practical questions, very questions on the field. But I'm not so much interested on that. You know, this is something that should be done by GWV, not for me, because it's like I will be working for them. So I'm more interested on the biology, and then, of course, but this is a possibility, of course. Yeah.
Thank you.
Okay. Thank you so much, Ismael.
You're welcome. Thank you very much for your interesting questions. I was astonished. It was very good questions.
Now, if you want to say something about Canalato.
Okay. Sorry. It's not that I didn't want, but I wanted to leave the time. So let me put the slide because I didn't prepare the slide actually for that. Let me show you something here. Can I? Okay, hold on. I don't want to do this. Okay. So is my presentation on there or no?
It's not full screen, but it's on.
Okay. Okay. So let me hold on.
You had something there, right?
That's my paper. Coming in. Here it is, Canalatan.
Oh, it's not appearing to us.
No?
No. I don't see
it. Is it Canalatan there? No.
No. It's
no.
Not for me. I can't see it. Can anyone
because okay. Let me do something. Okay, and then now, can I present again?
Yes, you can.
I need to find it.
I have to do something.
Okay, here it is. Okay, here. Okay, now. Now?
Yeah, we can see.
Yeah, okay. So yeah, Canalatan is basically a network. It's composed so far by 10 groups coming from different countries, from 8 countries. So we have 2 Spanish groups from Barcelona and our group in Madrid. We have 1 group from Costa Rica, the group of Cecilia Escoza in Uruguay, 1 group of Bolivia, and the group of Buenos Aires, as well as the group of Coimbra and the University of Mexico. And this is an initiative which aims basically to develop 3 axes: translational research on cannabinoid actions in the nervous system, and the training of researchers and professionals, and finally, social awareness, so information to the society. And basically, we are interested in investigating the consequences of either, for instance, CBD, and also some acid forms of cannabinoids, so CBDA, THCA, other acid forms of cannabinoids that do not act through CB1 on psychosis and anxiety. We've been investigating also with this company, Emerald Health Pharmaceuticals from the States, the use of chemically modified cannabinoids, CBD and CBG, on Huntington's disease. And also, there is a strong line of research on cannabinoids in epilepsy and mild cognitive impairment. And as part of the formation and training, we are developing different schools and courses about cannabinoids. There was this course organized by the people of Uruguay. We are having a course this week on cannabinoids and cancer in Spain. And we performed, we organized a clinical theoretical practical school on cannabinoids research in the CNES, which was proposed to be held in September this year in Brazil, actually. But it couldn't be, we couldn't have the course because of the COVID. So this course hopefully will be taking part, will be occurring next year in Brazil. Hopefully, I hope so. I cross my fingers. It was a lot of work to get the money and the speakers for it. And then finally, yeah, that's it. And that's it. So this is basically the CITER. And this is it. It's basically, we're basically promoting research stage between the groups of the lab, of the network. So our PhD and postdocs, they can get a different perspective of research and improve their training. It's more or less the idea.
So there are Brazilian groups as well?
So the only group so far is the group of Francisco Guimarães.
Mm-hmm.
Right. And now so the thing is, this is, I don't know how to say this. This is a very institutional call, let's say. So I'm still dealing with the process of understanding what the things I can do with the money, what the things I cannot do, and the interest of the institution. Actually, I have a meeting on Friday, so let's see if I can clarify. Because it's very, bureaucracy has been a pain. It's been so far in a pain for this month. So.
Okay, thanks.
Thank you, Cecilia.
Thank you very much, Ismael. So let's close the meeting. and I was gonna, let's see how we can do this. I don't know, wait a minute. I have to stop this because I wanted to call Claudia Benjamin, but I don't see how we can do this. Oops. Let's see. I think I'll have to leave.
Eu tô aqui, mas eu acho que tem muita gente, né? Eu tô aqui, mas...
Ah, então você que tá...
Só se as pessoas saírem, a gente fica ou abre uma outra sala, não sei. O Ismael vai ter tempo? Ele vai poder conversar com a gente?
A gente vai falar um pouquinho. Então, vamos parar o recording, Cláudia? Você que está tomando conta? Tá.
Ok.
Tá, pessoal, então. Boa reunião aí.
Tchau, gente.
Tchau. Vou encerrar aqui no YouTube também, tá?
Eu vou encerrar e vou te colocar como host, tá, Cecília?
Tá.
Ok. I haven't seen the...
Tá, obrigada.